Welcome back to the Brain Beat Prep by the Central Ohio Youth Neuroscience Coalition. Today we'll be talking about the second part of Chapter 2, Senses and Perception, and we'll be talking about the senses of hearing, smell, and taste. Last time we talked about vision, and next time we're going to talk about touch and pain, and then we'll be done with Chapter 2. So this chapter is going to be a lot of pathways and just basically memorization because we're going to talk about a lot of different senses. So first, let's talk about the sound pathway. So the sound pathway basically happens like this. The ear pinnae directs the sound into the ear canal, which directs the sound into the tympanic membrane or eardrum. And basically this eardrum, it vibrates, and the resulting vibrations go through these three bones, which are called the malleus, incus, and stapes. So what those bones do is that they amplify the sounds. They basically amplify the sound. They make it a little bit louder for the cochlea to hear it. So that, well, not really to hear it, but to like feel it. So then after that, the stapes pushes on this thing called the oval window, which is basically this membrane, and it separates the air-filled middle ear from the fluid-filled inner ear. So remember, this is air-filled, this is fluid-filled. You do not want to get that mixed up. And then in the fluid-filled inner ear, there's basically the cochlea. So there are three different parts of the ear: the outer, which is until the tympanic membrane; the middle ear, which is the tympanic membrane to the stapes; and then the inner ear, which basically just consists of the cochlea. So now that we finished the pathway leading up to the cochlea, let's talk about the cochlea itself. So within the cochlea, there's a membrane. It's called the basilar membrane, and it vibrates in different places at different frequencies. So let's draw this out. So basically, if this was a cochlea, then this would be the basilar membrane, and then this would be the oval window, and then on the other side would be the auditory nerve, which I'm just going to write as AN because we don't really need to know that until later. Well, next slide later. Well, actually, this slide later. So I guess we will need to know that, but that is besides the point. So within the cochlea, there's this membrane. It's called the basilar membrane. And then if you have a higher frequency note, so like say like over here versus like over here, higher frequency versus lower frequency is farther away from the oval window, and higher frequency is closer to the oval window. So if you have this type of note, which is a very high note, like if this was like a musical sheet, if you have this type of note, which is a very high note, somewhere on here would vibrate. And then if you have a low note, somewhere over here would vibrate. And so there are small hair cells, so let's draw those as well, that are on top of the basilar membrane. And when it vibrates, they actually bend against this other type of membrane, which is called the tectorial membrane. And so remember, don't get these two confused. They are very, very different. So the basilar membrane is where the hair cells sit, and then the tectorial membrane is where the hair cells bend against. So when they bend against this membrane, it causes these stereocilia, which is basically like these little hair-like openings. Well, yeah, like openings kind of to open ion channels, which leads to like an electrical signal. So basically it's converting sound to an electrical signal. And then, well, sound that has been converted into a fluid, like a fluid signal because the cochlea is filled with fluid, and then it's converted to an electrical signal. So what happens is that these axons of these hair cells make up the auditory nerve. So like right over here, that's what we were talking about earlier. And then the auditory nerve, it goes to the brainstem and thalamus, but it goes to different areas. And we're not really sure. We don't have like a specific area that it goes to, unlike the visual stem, like where we had the lateral geniculate nucleus or the LGN. And so eventually these signals end up at the auditory cortex, which is in the temporal lobe. So now let's talk about just some final words on hearing. So first of all, processing speech specifically, this is on the left side in like 90% of people, and then damage to the left auditory cortex, especially a place called Wernicke's areas, which we'll talk about more when we talk about language. It makes you unable to understand speech. So if you have any damage to the left auditory cortex, you could be left unable to understand speech because that's where speech is processed specifically. And then secondly, the loss of hair cells is basically like it underlies most of the hearing loss because these don't grow back. And so research is being done right now on how they function and develop so we can artificially grow them. So now let's talk about another pathway, which is smell. So in this pathway, the odors, this one is a little bit more simple, but in this one, odors enter the nose and then they bind to olfactory cells. So basically, if this is like a nose, they bind to olfactory cells that are located here. So like the odors would come up here and then bind over here. And then what happens is that these olfactory cells' axons, they cross through the skull and then they end up in the olfactory bulb. So if this is the brain and this is the olfactory bulbs right here, then they end up over here because they cross through the skull itself. So they do not go through the thalamus and brainstem like the other senses. Well, they do go through the brainstem, but they don't go through the thalamus at all. Until they reach the olfactory bulbs. This is not supposed to be lobes. These are actually the bulbs. So this will be updated on the PowerPoint that we upload on the Google Doc or the Google Drive. So it's just a typo. And then olfactory cells, they have cilia. So basically they have like these little hair-like cilia, and the cilia bind to the odors. And then each odor specifically has its own unique activity pattern depending on what cells it activates. So like if you have cells 1, 2, 3, and 4, perhaps odor 1, which smells like chocolate, might activate 1 and 2 and 3. Or like odor 2, which if you have the same 4 cells, odor 2, which smells like rotten eggs, might activate 1 and 2. So they activate different cells so that you have a unique activity pattern between all of this. So this pattern is actually transmitted to the olfactory bulbs, which are right here, and which are the place where the olfactory cells axons go, and then also the primary olfactory cortex, which is also located in the temporal lobes, like right here-ish. Oh, right here-ish. Like right there, somewhere around there. And then olfactory neurons are very, very special because they can be regenerated. So there are 2 things that smell is very, very important for. So first, you don't go through the thalamus before you get to the brain. And then second, the olfactory neurons can be regenerated. So if you're going to remember anything about smell, remember that. Even more than the pathways, you should probably remember those things because they are so, so important, because this is the only sense that does that. And lastly, let's talk about taste. So taste is sensed by taste buds, of course, which have many gustatory or taste cells within them. And so each cell is receptive to at least 1 out of 5 qualities, usually like more, or like they're receptive to like certain qualities more than others or something like that, of sweet, sour, bitter, salty, and umami, which is also known as savory. And so these signals actually go across 3 nerves to get all the way to the brain. They go across the pharyngeal nerve, the vagus nerve, and the facial nerve. So they go across a lot more nerves than the other ones. And then they go to the taste regions in the brainstem and the thalamus. We still we talked about with hearing, we don't have like a specific place they go. They kind of go like different places. So, and then eventually they end up in the frontal lobe's gustatory cortex and the insula, which is basically it's this middle side of the brain. Like if you draw a brain like this, you have your temporal lobe right here, right, and your parietal lobe right here. So if you open this up, then in the middle of this, like over here-ish, you have the insula. And so this is very important for taste, of course, and it's also really important for for taste. And then so all tastes, unless you might have heard that there's specific areas for certain tastes to be detected, but all tastes are detected across the tongue. That is a myth. There's no specific area for any part. All right, lastly, let's talk about some interactions between taste and smell. So these senses are very, very closely linked. So taste and smell actually become flavor. So you can't really taste things without smelling them. Like you can't do it very well. So if you've ever had a cold and you've been unable to smell, it's usually your food tastes more bland as well because you don't have that added sense of smell to contribute. And then so the signals of these senses actually converge in several parts of the brain because of that. So because they converge, then you have the flavor aspect of it essentially. And so some neurons, specifically in the inferior frontal lobe, which is basically like right here, like you have the front, you have the back, And then you have the central sulcus, which basically divides the frontal and the parietal lobes. And then right here would be like the inferior frontal, somewhere around there. And so they respond only to certain taste and smell combinations. So they don't respond to the taste itself or the smell itself. They only respond to the combination. And so because of this, we feel that the taste is more vivid a lot of the times when the smell matches it. And so that concludes the session. So please join us next time. We'll be talking about touch and pain.